Today we will be discussing hemodynamic consequences of coronary artery disease without symptoms of overt heart failure. The post-exercise muscle ischemia test will be used to analyze the metaboreflex of several subjects in order to examine the effects of coronary artery disease on the heart. First, we will be talking about coronary artery disease, or CAD. Coronary artery disease is the most prevalent form of cardiovascular disease. It involves the coronary arteries, or major blood vessels supplying the heart with blood, becoming damaged or diseased. Coronary artery disease affects both inotropism and the diastolic function of the heart. Inotropism encompasses the aspects that alter the force or energy involved with muscle contractions of the heart. Diastolic function of the heart refers to the portion of contraction in which relaxation occurs while the chambers of the heart are filling with blood. An important aspect about CAD is that hemodynamics, or the dynamics of blood flow, are dysregulated in patients who have coronary artery disease. This is relevant since it may eventually lead to overt heart failure. Next, we will look a little bit into the metaboreflex. The metaboreflex is a cardiovascular reflex that occurs within the heart following accumulation of metabolic byproducts produced during muscle contraction. Once these byproducts build up, the sympathetic nervous system becomes activated. This activation then triggers the cardiovascular reflex known as the metaboreflex within cardiac muscle. Measuring this metaboreflex actually uncovers important information about the functioning of the heart that is related to coronary artery disease. The test that is going to be used in the paper we will be discussing is called the post-exercise muscle ischemia test, or PEMI test. This test involves sitting in a chair for 3 minutes to establish a baseline, followed by 3 minutes of exercise involving dynamic hand gripping. Muscle ischemia was then measured on the hand performing the gripping, followed by a blood pressure reading being performed on that arm. This method was used here since it had proven successful in measuring metaboreflex recruitment in previous studies. During the PEMI test, the metaboreflex is isolated from other mechanisms that increase the discharge of blood flow during exercise. It is often used since the reserves that are present in healthy individuals, like cardiac preload, force of contraction, and afterload, can each be measured successfully. In a healthy person, their heart will adjust to the PEMI test by activating the inotropic and preload reserves until they are exhausted, and then will switch to SVR, or systemic vascular resistance. This test ultimately tells whether the person has a healthy response to exercise or not. Previous research shows that the consequences of CAD-induced overt heart failure during metaboreflex have already been studied. It has been found that an exaggerated SVR increment is found in response to the PEMI test, which can be attributed to impaired systolic and diastolic function. However, nothing is known about hemodynamic consequences of coronary artery disease without overt heart failure. Therefore, this study aims to establish whether the PEMI test can detect preclinical signs of myocardial dysfunction, eventually with the hopes of developing a diagnostic tool for myocardial dysfunction. The paper that we will be discussing is called Metaboreflex-Mediated Hemodynamic Abnormalities in Individuals with Coronary Artery Disease Without Overt Signs or Symptoms of Heart Failure. This paper is by Magnani et al. And was published in 2017. The goal of this paper is to investigate the effect of coronary artery disease without clinical signs of heart failure on cardiovascular adjustments during the metaboreflex activation. This was done using the PEMI method. The hypothesis that the researchers had said that the presence of preclinical systolic and/or diastolic dysfunction would reduce the possibility to achieve the target hemodynamic response by means of an SV-induced flow-mediated mechanism. This would lead to an exaggerated SVR response in these patients. As for the methods of this test, 12 men between the ages of 45 and 67 with coronary artery disease were selected to participate in this study. None of the patients had any symptoms of heart failure in the last 6 months prior to the study being performed. The second group involved a control group consisting of 11 men between the ages of 32 to 69 who were considered healthy and physically active. None of them had any sort of disease involving the heart or the lungs. All subjects performed a PEMI test to activate their metaboreflex, and their heart activity was measured as compared to their baseline. Each participant also did a control exercise recovery test, so the researchers had values to compare the results to from the PEMI test. In Figure 1, we are looking at absolute values during the post-exercise muscle ischemia test and the control exercise recovery test, and in response to the heart rate, stroke volume, and cardiac output in both the control group and the coronary artery disease group for each of these variables. A horizontal bracket here indicates the overall main effect of the groups, and this will be constant throughout the rest of the figures. There was a significant difference present here in the cardiac output from the control group to the coronary artery disease group. However, a significant difference was not seen in the heart rate or the stroke volume between the two groups. Here in Figure 2, we are looking at absolute values during the post-exercise muscle ischemia test and the control exercise recovery test once again in response to end diastolic volume, end systolic volume, and ejection fraction in both the control group and the coronary artery disease group. The groups have stayed constant throughout this figure, the last figure, and the next figure. Here, there was a significant difference between the two groups in regards to both the end diastolic volume as seen in A and the end systolic volume as seen in C, showing the impact of coronary artery disease on contraction of the heart. However, we did not see a difference in the ejection fraction between either of the groups during the PEMI test or the CER test. In Figure 3, we are looking at the absolute values during both tests once again in response to mean arterial pressure, systemic vascular resistance, and ventricular filling rate between the two groups. Here, there was a significant difference in the mean arterial pressure between the two groups using the PEMI test, but not with the systemic vascular resistance or ventricular filling rate for either of the two groups with either test. To summarize the results that were just presented in the figures, overall the researchers found that the coronary artery disease group reached a similar mean arterial blood pressure response to that of the control group during the PEMI test. However, the mechanism by which this arterial blood pressure response was achieved was actually different between the two groups. The coronary artery disease group achieved target blood pressure by increasing the systemic vascular resistance, while the control group increased the cardiac preload, leading to an enhanced stroke volume and enhanced cardiac output. Additionally, the researchers also found that the ventricular filling rate response was higher in the control group than it was in the coronary artery disease group during the PEMI test. Overall, this study confirms that diastolic function is pivotal for normal hemodynamics during the metaboreflex within the heart. If the diastolic function is not working properly, it is likely that the hemodynamics will be affected, and therefore the overall heart function will also be affected. This study also provides evidence that early signs of diastolic impairment from coronary artery disease can be detected by the metaboreflex test, which has important diagnostic implications, since we now believe that using this test, along with the PEMI test, can help indicate whether a person has issues with diastolic function that will lead to further heart problems. My two questions are, 1. Why do you think the mechanism by which the coronary artery disease group and the control group achieved the arterial blood pressure response was different? And 2, why was the post-exercise muscle ischemia or PEMI test a better option to test the metaboreflex than any sort of dynamic exercise? Here are my references, and thanks for listening.